Here's the second part of our Chapter 2 lecture. And this chapter, we are talking about two major topics when looking at cells. We're talking about the parts and components of a cell, which is what we discussed in our first lecture. And now the second lecture is going to talk about how cells make copies of themselves and how they divide. So this lecture is pretty straightforward. We're going to be talking about what's called the cell cycle. You can kind of think of it like the life cycle of a cell. And it's divided into two major parts. We'll talk about interphase, and then we'll talk about the actual division of cells itself, this idea called mitosis. And then finally we'll do our lecture review. Alright, so in the previous lecture we had talked about how we have all kinds of different cell types, hundreds of different cell types in our bodies, and those cells had specific jobs to do. So heart cells behaved like heart cells, and skin cells behaved like skin cells, and the reason that they knew how to function was because of the very particular genes that were turned on or off in those cells. Another critical function of living things in general is to be able to make a copy of yourself. So this would also include multicellular organisms. So not only do we need to be able to make an entire copy of ourselves through the process of reproduction, but we also need to make copies of ourselves because cells wear out. They have a very definite lifespan. They don't last forever. So we need to replace these cells on a constant basis. And some cells we replace more than others. So your skin cells, for example, you are constantly replacing skin cells because you're sloughing off thousands and thousands of skin cells every day. They just sort of come off of you. So you have to replace those skin cells on a very constant basis. Other cells you don't replace as often. So cells inside some of your organs like your heart or your liver, you certainly do need to replace those every once in a while, but really you're going to see those types of cells being replicated when there's damage done to those organs rather than on like a regular basis like some of our other cell types. So the cell cycle here is describing for us how a cell lives its life. And how a cell lives its life basically is in two different parts. What's referred to as interphase and then what's referred to as mitosis. And we're going to talk about each of these separately in this lecture. So just a quick overview of this idea of cell division. When a cell wants to divide, we need to make a copy of things. We need to replicate things. Specifically, we need to replicate the genetic information that tells us how to behave like a cell. So before cell division begins, we take our DNA and we condense it into those short linear chromosomes. So in the last lecture I talked about two different forms of DNA. I talked about chromatin and chromosomes. Chromatin was that loose, tangly web of DNA that is really good for accessing genes and for making proteins and things, but really difficult to make a copy of. Really difficult to move around a cell. When we go into the process of cell division, we want our chromosomes to be easy to move around because we're going to move copies of these all around the cell. So what we do is we turn them into these short linear chromosomes. So keep in mind that there's an entire process by which we're replicating all of our DNA. And actually, we'll go through how DNA is replicated in Chapter 9, which is a chapter that we're going to jump to very shortly here in the next module. So just a little bit of terminology. When we make copies of chromosomes, we refer to them with a new name. So we don't necessarily call them chromosomes anymore. We begin to refer to them as sister chromatids. So the sisters are connected to each other at a point which is called a centromere. So let's take a look at this. So here's our chromatin. This is what's hanging out in your nucleus. 98% of the time your cell is alive. It's just hanging out like this. All of the genes are accessible. We can make proteins. We can do all the stuff that a cell needs to do. But for a very short period of time during the cell cycle, we need to make a copy of the cell. And in order to do that, we need to make a copy of all of this information and then move it around the cell. So what we're going to do is we're going to take this tangled web mass, and we're going to replicate it. Okay, so we're going to make an entire copy of it, and then we turn it into this nice little X-shaped replicated chromosomes. So replicated chromosomes are always easy to identify because they look like the letter X. Each half of this, right, is a single copy of the genetic information that we need. Okay, so we have 2 copies right here, and they're held together at this little point called the centromere. During the process of cellular replication, what we're going to do is we're going to move these chromosomes and separate the sister chromatids from each other. And there's a very precise way that we happen to do this. The division of the nuclear contents actually is a process we refer to as mitosis. And it's referred to as an asexual division because technically single-celled organisms can actually undergo a process very similar to mitosis in order to make copies of themselves and replicate. And you don't need a partner to do that, so that's why we call it an asexual form of replication. Again, it's part of this overall cell cycle, which was the life cycle of the cell. And the whole goal of mitosis is to make an identical copy of the original cell. We want absolutely everything to be the same. Nothing should change, okay? Same genes, same organelles, same DNA, same everything. Everything has to be precisely the same. All right, so before we can get to mitosis, a cell needs to be ready to do that. So in order to be ready to do that, there are several steps that we have to take. And those steps are basically set up into interphase, mitosis, and cytokinesis. So we're going to talk about all 3 of those today, but you can kind of think of it like this way. So in our diagram here, here's our cell. During interphase, we're going to make a copy of all the genetic information. During mitosis, we're going to split that DNA equally between 2 cells. And then finally, during cytokinesis, we actually pinch the cells into 2 so that we have 2 distinct cells. So that's a general overview of what we're going to talk about today. All right, so interphase again is when a cell is being a regular cell. A heart cell is doing what a heart cell does, and a liver cell is doing what a liver cell does. They're carrying out their normal functions. There are 3 stages of interphase, 2 gap phases, gap 1 and gap 2, and in between them is the synthesis phase. So G stands for gap, S stands for synthesis. What's going on? Well, basically the cell is growing. We are replicating organelles. The cell is getting larger in size. So a cell has to be a certain size before it's able to replicate. Then we get to the synthesis phase where we copy all of our genetic information and we attach our sister chromatids together by centromeres. And then last but not least is gap 2 phase where we make all of the proteins necessary for us to go through our division stage, our mitotic stage. Okay, let's do a quick comprehension check. What is the purpose of interphase? Is it to allow the cell to grow? Is it to carry out normal cell functions? To replicate DNA? Or all of the above? It's all of the above. We are doing all of these things in interphase in order to get ourselves ready in order to divide. All right, so division, formerly called mitosis. The whole purpose, again, is to separate those sister chromatids so that each new cell has a complete set of chromosomes. There are 4 phases in mitosis. An easy way to remember it is PMET. Prophase, metaphase, anaphase, telophase. Each phase is defined by what the chromosomes are doing and what they look like in each phase. So, we will start off with prophase. We have chromosomes that are condensing. Okay, so we have all that replicated information. Now we're going to make these short, fat chromosomes and make them visible. Certain cytoskeleton features. Remember how we talked about the cytoskeleton in the last lecture? We talked about microtubules and microfilaments. Microtubules are special cytoskeleton features that form basically an anchor point at the poles of our cells, which are kind of the ends of the cells. It's an anchor point that we're going to use. The nuclear envelope disintegrates. Okay, so remember in our cell we had an envelope that was surrounding that nucleus, which was regulating things going in and out, accessing that genetic information. Well, we're going to get rid of that so we can move our replicated chromosomes all over the place. Next is metaphase. In metaphase, those replicated chromosomes are going to line up across the equator, or the middle of the cell. Those microtubules that we just formed are going to attach to those centromeres, and we're going to create these things called, basically, they're a type of, those microtubules are referred to as spindle fibers. All right, so 2 phases of prophase, or 2 views of prophase, excuse me. This is an actual cell, if you were to take a look under a microscope. And this is just our drawing, okay? So we've got our chromosomes are visible. Our nuclear envelope is going to disintegrate. These microtubules that I mentioned to you are actually called, we refer to them as spindle fibers. Okay, so the spindle fibers start to actually come out of the poles here, and these centrioles, again, is just another form of microtubules, all of these cytoskeletal elements. Next is metaphase. So what you'll notice in metaphase now is we have our equator of our cell, and you will notice that all of our chromosomes are lining up very, very nicely right along the equator. And what you'll also notice, and you can see actually very well in this picture here, see these little fine, what look like filaments attaching to all of these sister chromatids? Okay, those are those microtubules. Those are those, what we call them spindle fibers. That's the technical name that we give them. And what they're doing is they are actually physically moving around these sister chromatids all around the cell. So that was prophase and metaphase. Let's talk about anaphase and telophase next. In anaphase, we're actually physically pulling the sister chromatids apart from each other. Okay, so we're going to use those microtubules, those spindle fibers, to pull the sister chromatids apart towards the poles. And then in telophase, we're going to take those two distinct groups of chromosomes now, and we're going to put the nuclear envelope back around them. And the chromosomes are going to go back to their uncondensed form, to that chromatin that we talked about. So this is what it looks like again in the cells. We have the sister chromatids are pulled apart from each other. They're no longer joined to each other. We are pulling them opposite directions toward either end of the cell. And then finally what we see is we have, again, what look like two distinct cells now. Okay, so we've got a nuclear envelope here forming around this set of chromosomes, and we have a nuclear envelope forming around this set of chromosomes. And then in the middle, we're doing something called furrowing. Okay, furrowing is, you can think of threads being strung around the middle, again, more microfilaments and microtubules. They're actually being thread around what was one giant cell, and they're actually pulling, and they're making the cell membrane pinch into two. And that pinching into two is technically a process called cytokinesis. And in cytokinesis, we divide the cytoplasm up between those two cells. And the organelles are sort of evenly distributed between what we're calling our two new daughter cells. So that terminology, daughter cells, is just the two cells that we end up with when we're done with mitosis. All right, so then what happens? Okay, so it's all over. Mitosis is over, cytokinesis is over, and then the cells go right back into interphase. They enter into the gap1 phase of interphase, and they repeat that cell cycle. And again, how quickly they move through the cell cycle depends on what type of a cell they are. Skin cells, which are constantly, almost constantly dividing because, again, you slough off and lose so many skin cells on a regular basis, they go through that cycle very quickly. Whereas other cells in other parts of your body go through it much more slowly because you don't need to replace those cells on a constant basis. And again, rates vary depending on different tissues and at different times, and it's all regulated by this internal clock. All right, so what prevents a cell from just dividing whenever it wants, however it wants, as many times as it wants? Well, there's a whole host of checkpoints that actually are in place to make sure that everything goes in the correct sequence, and there are both internal and external factors that are involved in this. What's interesting is a cell that has lost control of the cell cycle, that is dividing uncontrollably, actually is the beginning of what we refer to as the disease of cancer. So cancers are masses of cells that begin to grow out of control, and they have lost control of the cell cycle, basically. It's because the checkpoints that are there in place to prevent them from doing this have failed. So we have all kinds of checkpoints here. So we have checkpoints making sure that we have, if the size of the cell is big enough, if the DNA has been damaged at all, we make sure and we go back through and we can correct all of those mistakes that are made. We take a look and make sure that the spindle is there correctly, is it attaching to the chromosomes. So again, all of these checkpoints are in place to make sure that a cell doesn't go through the cycle too hastily. But sometimes mutations occur. Mutations are errors in our DNA that prevent a gene from doing its job. So the genes that are supposed to do their jobs here to ensure that all of these checkpoints are actually being met, sometimes they fail. And when you have multiple failures of these checkpoints, you begin to see cells that begin to grow uncontrollably. And when they go uncontrollably, they can form a mass. And that mass, again, can eventually potentially lead to cancer. We'll talk a little bit more about cancer a little bit later in another lecture. We'll talk more about it, but that's sort of the beginnings of that disease, is when you have cells that are dividing uncontrollably. Last thing I wanted to mention real quick are something called telomeres. And your telomeres are located at the ends of your chromosomes, and they're kind of like a protective tip, if you want to think of them that way. So think of the plastic tip on the ends of your shoelaces, and it prevents your shoelaces from coming unraveled. That's what telomeres do. So they are these little protective tips on the ends of your chromosomes, and it's basically thousands of repeats of this 6-base sequence of DNA. And every time you divide, you lose some of those bases, anywhere from 50 to 200 of those every time the cell divides. So then what happens is, over time, as your telomeres get shorter and shorter and shorter, it's a signal to your cells to stop making copies of that cell. Because basically that cell is getting old, and it's getting tired, and there's probably more mistakes than we want to start making copies of. So it's a kind of an internal way to stop that whole process of division. Certain cells, sperm, egg, bone marrow, and cancer cells, produce something called telomerase that prevents the shortening of those telomeres. So remember those cancer cells that I talked about that said they divide uncontrollably? Well, they should stop after their telomeres are gone, right? Nope, there's a protein called telomerase that actually prevents the shortening of those telomeres, that allows them to divide as many times as they want. All right, last comprehension check. In which phase of mitosis do the chromosomes align at the equator? Is it prophase, metaphase, anaphase, or telophase? Metaphase. You can think of metaphase middle. That's kind of a good way to remember metaphase. Chromosomes are in the middle. Here is the review slide, all of the important terms and concepts you need to know. Make sure on Blackboard you check out some additional resources that I have posted that are particularly helpful for remembering the phases and steps of mitosis.